So for the first talk for today, I'm going to welcome Professor Judi Allen. So she's the lead for Immunomatrix. So Judi started in science with her PhD at the University of California in Berkeley. She then did her postdoc studies at Imperial before moving to Edinburgh to set up her own research lab. And it was in Edinburgh where Judi's lab was one of the first labs to show macrophage activation in the context of type 2 immunity in vivo. And Judi has recently been awarded a prestigious EMBO membership. So Judi has a special interest in macrophage activation and tissue repair, and in particular in the context of helminth infection. So Judi's going to talk to us today about the research going on within the Immunomatrix theme.
Thanks, Lizzie. Thanks, everyone. I feel a little bit of a fraud. I'm the leader on Immunomatrix, and I'm discovering, especially in the past couple of days, how little I actually know about Matrix. But what I would say is that in the 2 years I've been in Manchester, I've become a completely evangelized about the importance of Matrix, to the point that I've now become evangelical. As I was just telling somebody, I'm a missionary's daughter. So I've become evangelical about the role of Matrix in everything, and that we really, if we're going to harness complexity, we have to embrace complexity, and understanding the complexity of the matrix is part of what we're going to have to do if we're really going to understand the immune system and how it interacts with cells. So the matrix in our body is 70% of our body mass. As Carl Cadler, the director of the Wellcome Trust Center for Matrix Biology here likes to remind people, is if you didn't have matrix in your body, you would just be a little pile of cells on the floor. So we really are essentially matrix. And there's all sorts of forms of matrix. There's the mucous matrix, which is something we have great strength in here, which I think is quite unique to Manchester. You then have these cells. They're kind of important. And then underlying them, you have the basement membrane. And the basement membrane underpins all these tissues. And in fact, destruction of the basement membrane is a really common feature of lots and lots of autoimmune diseases. You then have structures like elastins that give us tissue flexibility. You then have the collagens that give your body tensile strength. And actually in your little brochure, which I'm sure you've all now read through very carefully, it says that 30% of our body mass is matrix. That was an error on my part. 30% of our body mass is collagen, and collagen alone. And Carl corrected me on that today. And then underneath many of our tissues, we have these huge areas of hydrated gels, which are the glycosaminoglycans and the proteoglycans, which hold water, but also give us that ability to resist gravity and protect all our tissues. And then you heard the great talk from John about how good we are at understanding the barrier. But once again, I would have to say to people, if we're really going to understand barrier immunology, we have to put everything in the context of matrix, because our barriers are matrix. Cells are only a very small part of that. And I've already described those aspects, but organisms have to move through that matrix. Injury breaks that matrix. Autoimmunity destroys that matrix. So there are all sorts of aspects of the barrier that really I don't think can be studied without considering the matrix. So the disruption of the matrix is the direct or indirect cause of most major chronic diseases. 45% of all deaths can be attributed to fibrosis. So fibrosis, of course, is the excessive deposition of matrix. Of course, one of the things I sort of failed to mention is that as we age, which is an important part of understanding immunology, our matrix is changing all the time. So it's also a very dynamic process. Cardiovascular disease obviously has a role in terms of heart disease. You have the expansion of matrix, but also our blood vessels are matrix, and they're changing all the time. Cancer, I think without question, we understand an increasing area of urgency and interest is that the tumor microenvironment is predominantly matrix, or cells that make matrix. And then, of course, both arthritis and asthma, also huge strengths here, are diseases in which the matrix is being remodeled. That is actually the problem, the underlying problem in those diseases. So in terms of putting this in the context of immunology, I think it's really important to understand that immune responses modulate the matrix. Matrix is not inert. So cytokines regulate the deposition and composition, for example, of the mucus, and that once again, I've said that's a strength here. People like Dave Thornton, and you'll hear a little bit more about it from Richard Grensis. My favorite cytokine, which you'll also hear more about, IL-13, drives matrix production, but it also changes the nature and the context of the mucins that make up the matrix. So the immune system is not just changing the quantity of the matrix, it's changing its quality. We also know that immune responses can completely change your collagen architecture. I'll talk about this a little bit later, but the size of collagen fibers are regulated by particular enzymes that can be changed by cytokines. And then these proteoglycans and glycosaminoglycans that make up the interstitial spaces can also, their expression is critical to interactions between all sorts of cells, and that is also regulated by the immune system. And of course, the reverse is true. The matrix, this is an animation, I didn't realize it, the matrix also modulates the immune response. So one thing that's well known in many fields, and I think is becoming more apparent in immunology, is the stiffness of tissue regulates cell function and cell fate. And in my area of macrophage biology, it's very clear that how stiff the interaction is with the tissue will determine its polarization state, or at least regulate its polarization state. So all that work done in vitro is missing out on a really critical component of how the matrix is modulating our cellular functions. And then probably the best known area, if one were to think about immunomatrix, is the fact that these glycosaminoglycans are repositories for chemokines and cytokines. So, chemokines have to let a trail along there on these tissues, but also because not seeing that. Because this matrix of sugars is changing all the time, it's also going to be regulating the immune response as it holds on to growth factors, chemokines, and so if that gets disrupted by injury, inflammation, or infection, that's also going to alter the immune response. And I think that's a major fundamental aspect. We tend to think about cytokines in isolation, but most of them are actually bound up in matrix. There we go. So the major themes here, like I say, I'm a little bit obsessed with IL-13, so I put it at the top. But IL-13, fascinating to me, regulates all aspects of matrix, and particularly it regulates collagen turnover and deposition, and it also regulates mucus, as I already mentioned. Part of the way IL-13 does these things is by it also acts on TGF-beta, and you heard from Mark about that. But one of the things, another focus here, particularly through Mark, is not only the ability of TGF-beta to regulate the matrix, but more and more data has shown that it is the matrix itself that has the molecules on it that can activate TGF-beta. Another area that I think is, as I mentioned, is the binding of these molecules to glycosaminoglycans, and we've just recruited a new fellow who has really showing that particular chemokines are able to interact with the matrix to actually change the structure of the glycosaminoglycans in such a way to allow cells to attach to the tissue. So it's actually the chemokines changing the matrix rather than just binding to it. We have another big interest in circadian, which you've also heard about, and we also know that not only does our matrix change as we age and with time, it's actually turning over every day, and that's controlled by the clock, and we know that the cytokines can modulate that. And so obviously if you don't sleep well, you don't remake your collagen every night, and we know that the immune response is a major player in that part. There's a really growing and exciting area here with the nursing faculty on understanding complex wounds, and I think we can really contribute to that with our understanding of how the immune system controls matrix. And a major theme really as a part of the Wellcome Trust Center for cell matrix, which is obviously a real fantastic interaction here between us, is an understanding that dysregulation of all these processes will lead to fibrosis, which, as I said, is a major cause of death, probably the major cause of death worldwide, certainly in the Western world. So I'm going to try and tell a bit of the rest of the talk, tell a bit of a story that's close to my own heart that I hope will illustrate a number of things. It will illustrate a strength here in in Manchester of parasite immunology, where and you'll hear more about this from Richard, how these large extracellular metazoan parasites have evolved with our immune system. And in studying those interactions, we're learning about a lot of fundamental biology and immunology. And I've had this, for many years, have been very, very interested in IL-4 and IL-13, and I'm just beginning, because of these interactions here with the matrix biology people, to begin to understand how the activation of macrophages by 4 and 13 is affecting the matrix. And I'm going to tell a story that doesn't have an ending yet, because the ending will only come when we've really, really done the experiments with the matrix biology people here. So one of the things we know is that genes associated with tissue repair and matrix remodeling are highly expressed in response to IL-4 and IL-13 signaling. IL-13, along with TGF-beta, are the two most pro-fibrotic cytokines in our body. And I think we know a lot about how TGF-beta is pro-fibrotic. We know a lot less about how IL-13 is pro-fibrotic, although in part it acts via TGF-beta. So why are IL-4 and IL-13, which is, without question, the immune response we mount to these large extracellular migrating parasites, why is that so strongly associated with tissue repair and remodeling? And I think if we fundamentally understand the biology of these parasites, it becomes pretty obvious. They're very tissue damaging. And I'm going to briefly give you the life cycle of the hookworm parasites, which affect about 1/3 of the human population and a much higher level of animal population out there. And it's also, I always tell this, I've always said I'm a missionary's daughter. I was not allowed to go barefoot as a child because of this parasite. So what happens is the parasite crawls up a blade of grass. It senses your body's heat and CO2. It then burrows through your skin using proteolytic enzymes. It gets into your bloodstream. It gets swept from the bloodstream to the lung. It then bursts from the blood vessels into the alveoli of the lung, causing quite serious hemorrhaging, which needs rapid repair. It then molts. It then crawls up your trachea. You cough. You swallow. It ends up in your small intestines, where it sucks your blood. It's biting your gut wall every 2 to 3 minutes. And if you don't repair that damage instantly, you'll die of sepsis. So I think it's pretty easy to understand why infection with parasites is important, why wound repair and tissue remodeling is an important part of type 2 immunity. So when you have a hookworm parasite like that, you have this blood vessel. It's got a basement membrane. It's then got interstitial space with aminoglycans, and then you've got the endothelial cell lining, and the parasite has to come from the blood vessel into that alveoli, and it does a fair amount of serious damage and causes hemorrhage when it does that. And that barrier is now disrupted and needs to be rapidly repaired to prevent more bleeding and to prevent fibroblasts and other cells that shouldn't be in the alveoli from escaping out and causing fibrosis. So what we know is that IL-4 and IL-13 accelerate tissue repair. That makes it happen faster, which is probably a good thing, but that has the potential to lead to scar tissue. And that animals that can't respond to IL-4 or IL-13, the IL-4 receptor knockouts, repair their wounds more slowly, and they also exhibit enhanced hemorrhaging. So one of the molecules that we've been focusing on a long time is the most abundant protein that is produced in the context of IL-4 receptor signaling. And it's made predominantly by macrophages, but other cell types as well. And it's a molecule called YM1. It's associated with almost any injury response. It's a chitinase-like protein. It's part of a large family of proteins, but we really don't know what it does. What we do know is that it binds these glycosaminoglycans. So you have the endothelial cells, the basement membrane, all these things are populated with molecules that are expressing a variety, heparan sulfate and other glycosaminoglycans, and we know YM1 can bind to those, and we assume that's part of its function, but we really don't know how. And why does this even matter? Because actually YM1 is a mouse molecule. Well, YM1 we believe is an excellent model for a human molecule called YKL-40, where serum and sputum levels correlate with disease severity in asthma and COPD. Plasma levels are really, they're basic. If you have high YKL-40 in your bloodstream, you're probably one of those people who's going to die. So it's really not a good prognostic marker for many fibrotic diseases, and well, you'll find the most manuscripts are in cancers. It's a biomarker for poor prognosis in cancer. So it's a very highly expressed molecule in humans as well as mice, but we really don't know what it does. So what Tara Sutherland, who's a fellow here, wanted to ask the question is, does YM1 contribute to tissue repair? So we were using a model of hookworm called Nipostrongylus brasiliensis, a model in which this IL-4 receptor signaling is known to be needed for the rapid repair that I talked about. And so we wanted to ask what was the role of YM1, this chitinase-like protein, in this system. So YM1 is, and another closely related IL-4 receptor-dependent molecule, RELM-alpha, are expressed very highly in the lung in this infection model. So what happens in the infection model is the larvae are in the lung by 24 hours, up and out the throat and in your intestine by 48 hours. But as the lung is very highly damaged and you have this lung injury, you get a big increase in the Th2 type immune response that we know is necessary to repair that damage. And this is just a picture. In red is YM1 and in green is RELM-alpha. And so what Tara did was to block YM1 at the time when it was the amount of YM1 is really increasing. And it was very clear immediately that you actually need YM1 in order to get the lung repair, because if you block it, trust me, if you can't look very closely, is the lung is not repairing as well if you've blocked YM1. And critically, and I'll come back to this, is you also have excessive microbleeding, which is also a key feature of what we see in an IL-4 receptor deficient animal. So in an animal that can't respond to IL-4 or IL-13, we see this excessive bleeding. And we see that in a wild-type animal where we've blocked YM1. What I think is more remarkable, and is actually a very recent experiment, if Tara delivers YM1 intranasally to a mouse that's had its lung damaged by this worm, in an IL-4 receptor deficient animal, that repair doesn't happen very fast or very well. Delivery of YM1 alone is sufficient to rescue that phenotype. So this single molecule is sufficient to rescue the failure to repair that you see in an IL-4 receptor deficient animal. And the other thing that she noticed, which was very striking and very big surprise for us, because we've been working on these two molecules for a long time, is that YM1 drives this epithelial RELM-alpha expression. Because if you block it or deliver it, which I'm not showing here, it's affecting it. So if you block it, you don't get the RELM-alpha expression. In an IL-4 receptor deficient animal, if you deliver YM1, you now get RELM-alpha expression. So one of the obvious possibilities here was that YM1 is acting via RELM-alpha. And we had that was a great discovery, because through a collaboration with Sabine Emming in Cologne, she had shown important roles for RELM-alpha in tissue repair through collagen remodeling. So what Sabine had shown is in a skin biopsy model, if you lack IL-4 receptor signaling, your collagen bundles are very heterogeneous rather than homogeneous. And she was able to show that that was due to RELM-alpha. She could deliver RELM-alpha to an IL-4 receptor deficient animal and rescue that phenotype. And what RELM-alpha does is it induces an enzyme called lysyl hydroxylase 2 that is involved in cross-linking collagen to make it give it mechanical strength. So you don't normally find this enzyme in the skin or in the lung, which is where we've now found it, but when you do, it's involved typically in scar tissue or making things stronger. The other thing lysyl hydroxylase is important for, this particular one, is for forming blood vessels. So in a way, it also allowed us to explain why we get this accessing bleeding phenotype, is that you need this enzyme that is being driven by RELM-alpha in order to form the blood vessels properly during angiogenesis. And so we know that RELM-alpha is driving this. So we had all this data from this paper where Tara and I had worked with Sabine, and now Tara was able to show that the same process is happening in the lung. And I won't show you all the data, but just to summarize that, we know that myeloid cells activated via IL-4 facilitate rapid repair, and we think, and I would really strongly emphasize that this is just the tip of the iceberg in how type 2 immunity is controlling matrix regulation and deposition, and I think there's a lot more to learn, but that IL-13 induces YM1, that induces RELM-alpha production by epithelial cells, which drives fibroblasts to produce this lysyl hydroxylase that's involved in the appropriate cross-linking that leads to mechanical and biochemical stability of the tissue with these collagens, but also almost certainly is important in scar formation, so that leading to the importance of understanding this in fibrosis. fibrosis. And of course, all sorts of implications to cancer where these same sorts of processes are going on in the tumor microenvironment. So just to end, as I say, the story has not ended. What we know is that YM1 is a chitinase-like protein that's produced in vast quantities during injury, and which regulates immunity and tissue repair, and it binds extracellular matrix. So it binds GAGs, but that's all we know. And one of the huge opportunities for us here is to have this fantastic Wellcome Center that really understands matrix biology in a way that I really completely don't. And so in collaboration with Tony Day, we're now going to begin to define what those interactions are, because these glycosaminoglycans are different in every tissue. They're changing every day in our bodies. They change during injury. They change with age. So is YM1 or these other chitinase-like proteins binding to specific glycosaminoglycans? And if they are, can we interrupt that function and perhaps develop tools not only to unravel function, but potentially for therapy as well? And the long-term impacts for therapy are very big, because many of you might know dupilumab is now very much in the clinic for treating atopic dermatitis. So there are a number of molecules that are out there approved within the UK for use in the clinic to block IL-13, so maybe would have roles in fibrosis. I think there's a lot to study there. But another idea that's put forward a lot is to use local delivery of IL-13 to accelerate repair. To be honest, I don't think any of these are very good ideas. I think we should probably focus on molecules like YM1 itself and interact with how it's changing that tissue. So I will end there just with this beginning slide again to say I don't have a list of all the people who are in the Center Institute who are in the Matrix group, but if you're not in it, you should be, because Matrix is underlying everything you do. So thanks very much.